Okay, in the first part of this lecture series, we talked about the open reading frame, the part of the gene, a eukaryotic gene that we think of as the main part that will eventually make the mature message, as we call it, that will end up making protein, right? That'll be part of ultimately of the translational process. And we described the fact that there were introns and exons and some of the characteristics of those. We all now want to talk about where this green arrow is pointing, some of the regulatory sequence, some of the what's called the upstream or the downstream regulatory areas. Because imagine you've got 20,000 genes. Uh, those genes are found in every cell in your body that's somatic cells, but you don't need all 20. In fact, you may only need a few hundred if you've got, muscle or something, or maybe a few thousand if you've got a more complex system, but you certainly don't need all 20,000 turned on at one time. So many of them have to be turned on, turned off. But if you ingest some food, those cells that need to secrete something need to turn on the genes for that. So you may want to turn genes on and turn off. So there may be permanent on-off switches as well as momentary on-off switches that you can turn on. So we're going to talk about the regulatory area. Now we're going to talk about a very general view of this at this point. Later on in the semester when we talk about gene regulation, we'll come back and hit on some of these and be a bit more specific about some examples of what are going on. Here, again, we're just building a consensus, right? So we're talking about this consensus structure of eukaryotic genes, kind of a general idea of the basic elements that have to be there for it to work. And first off, we've got to have a unit that's transcribable, right? So we've got to have a transcriptional unit. Some area that we now know usually contains introns and exons, so areas that will eventually end up outside of the nucleus, and other areas that will be cut out in scenes, if you will, of a movie that will remain in. So now we want to look at primarily at this regulatory area that we find up here. It's known as the 5' flanking region regulatory area here. Okay, and it's a binding site for proteins. When we say regulation, generally we mean either RNA will bind there, or in cases of turning things on and off, proteins. So some genes, actually, we read the code, we transcribe it, we modify the code so that we splice out things that we won't want. We go out and we go to translation out in the cytoplasm, and then we cycle that back into the nucleus to actually sit down and do things on these regulatory areas. So these proteins, some genes just go for proteins that go and interact with other genes, things, okay? Now we're going to talk right now about an area that's generally called the promoter region. It's an area that can be anywhere from a few hundred to a few thousand bases of area above the gene itself, okay? So we would say that this is some of that area of, well, what is the junk? Well, this is the junk. It's not junk at all. It's actually very important code. While it doesn't produce phenotype directly, it controls the production of phenotype. I'm going to talk about just a couple of areas that are found inside of the promoter, just to give you an idea of things that may find there. Generally, one is called the Tata box. The other one we'll call the cat box. Because these represent some motifs that are common there. And then we'll close out and we'll talk about what are called enhancers. Enhancers are pieces of DNA where things can lock down, those things being either RNA or proteins lock down or release that impact and control whether this is functioning at all or not. And then there are GC-rich regions down on the 3' end down here, and also, of course, some start codes and other things. Now, this region on this end, I'm not really going to talk about much, this downstream region, but it is very important. And in fact, a lot of regulation is actually downstream. And that's because when you open up the DNA to read the code, there's a lot of folding and other things that can occur with that single-stranded DNA. So sometimes the downstream region plays as much as an important role in promoting as the upstream region for certain genes. All right, just some definitions and to give you an idea. The Tata box here is a highly conserved sequence. It serves as one of the initial binding sites for RNA pol. Now, RNA pol doesn't particularly link there, but RNA polymerase, pol, right? We're going to make RNA, start transcription, but it's one of the areas where it groups up, as we'll see when we talk about transcription, all the proteins needed to sort of make the process work. So it's a recognition site for where RNA pol ought to start and be brought in to start to function, okay? In that system, because it's a conglomerate of proteins that have to come together to actually make a transcription work, It functions basically as an on-off switch. Okay, the TATA box is either transcription on or transcription off. If you block the transcription of the TATA box, which some proteins do to turn a gene off, then the switch is off. If you want to actually read that code just downstream from there, then you activate the TATA box. So literally it's an on-off switch. Okay, so it's an area where we have a start, right? It's not the start of the gene, which is ATG, it's start of the actual transcription process. So here's my little mouse, my little beach mouse, and here's P62 sequestosome that we do work with. So where is all that in here is the question. You know, this is the promoter. We've sequenced the promoter. We know that the gene is starting down here somewhere, and somewhere up in here is a TATA box. Well, it turns out that the TATA box is about here. It's about 765 or so bases up from the start of the gene. There's the official start of the gene, and there's TATA. Now, a lot of this gets transcribed up here, but it doesn't get used. It gets cut out. But eventually, that will be the first code that's used to make up the P62 protein. But TATA is found right up in here. That's a classic TATA area. Okay. All right, now so you've got on off, right? Well, in your car, you know you've got an on off switch of some kind, a button that you push, or you've got a key that you insert and turn it on, start it up. Now it's running, but how fast it needs to run and all that needs to be controlled. So there needs to be something kind of equivalent to your accelerator, and that's known as a cat box. Now, the cat boxes, that's a well, there are specific codes that represent cat boxes. In fact, a very common code in mammals and others is this GGCCAATC. That's a consensus code for a cat box. There are many other things that function like cat boxes. The point of a cat box is saying it here is that I want you to know that there are accelerators. Okay, and cat boxes control how fast this gene may be copied, because sometimes you need multiple copies of the same gene, so you want it to keep going through the process, or just the rate. You don't need it for a while, then you can slow it down to make that one copy, or maybe you can speed it up to make the one copy. So it acts very much like an accelerator. Okay, and there are very much cat boxes. There are things that have this consensus code, just like you saw the TATA code here, but they don't necessarily have that particular code, but they still function in the same way. Okay, going back to my little P62 here, okay, here's the gene off down here again. Here's TATA, right? And actually, here's a version of what the cat box looks like. This one and this one actually help control some of the speed at which this gene happens to be functioning. We know that because we can mutate some of these areas so they don't work, and you can see what product you get from that. So that's the way actually you figure it out, is you go in and you forcibly mutate these so that they quit working, and then you look to see what product you get given under normal conditions. And then if you go look at other genes, there are all sorts of areas that function as part of this whole process of initiating transcription, controlling whether it happens at all or not. Here's the cat box for this one right here. Here's TATA box, very formally TATA box. But there's all sorts of other areas where proteins lock down, and either they enhance the process of transcription or they inhibit the process of transcription. And it's all found in these promoter regions. All right, the last thing of these I want to talk about are called enhancers. Enhancers, notice these little lines right here, that represents the fact that they can be many hundreds, if not thousands of bases away from the actual transcriptional unit that you're dealing with. In fact, there may be 3, 4, 5 genes in between this gene we're working with and the enhancer, and it may control multiple ones of those. So enhancers are thought of as specific DNA sequences. They're nothing but sequences, and they are shared between genes, and they coordinate the activity of genes. Right, so they're actually a coordinator. So they're the first things that kind of activate to help make all of this function. And again, when we talk about gene regulation, we'll spend quite a bit of time talking about how enhancers work. But for our purposes, let's just think about the system here, where we want to have expression of some genes, okay, but we want them to be expressed differently in maybe the brain and just differently than, say, in this case, in the limbs, right? We want to have, that's what enhancers do. This little green box right here and this little blue box right here are enhancers. If you're in the brain, certain elements come in, proteins and other things that say, no, I want to use this enhancer. And when I use that enhancer, it uses maybe this particular gene here, but not this one, and then this one. And there are things we'll see called insulators and all sorts of things that play a role there. Okay? In the limbs, maybe in the muscle or limbs or wherever, you use a different promoter, and in fact may allow you to do different combination of genes to be output there. So it says, yeah, I kind of need some similar stuff here, maybe to make it, mitochondrial function work or energy flow or whatever. But in the brain, I need this type of protein produced. And in muscle or other appendages, I need this type of muscle produced. So it functions as to help with that. And we'll talk a lot more about these things called enhancers. Again, they're far removed from the genes you're interested in. Maybe that's one gene, that's another, and there's several in between. And we can turn that gene on and skip these two and turn that one or whatever. There's lots of different options for how these work. Okay. All right. So going back to where we are, we have a consensus sequence now. We have our transcriptional unit with introns and exons. We have an on-off switch that we'll call a cat box or a TATA box here. We have an accelerator we'll call a cat box or something similar to it. And then we have these other structures that are upstream called enhancers that control the system. Right? Your book puts it pretty much like this. It calls RNA coding region. That's the gene or transcriptional unit. This is called upstream. This is called downstream because of the way the message is formed. Okay. Now, last little thing I want to do is just take you off and show you, okay, here's the real world though, all right? In the real world, remember you've got double-stranded DNA, right? It's anti-parallel. Well, turns out that you can use one strand or you can use the other. Either one can produce message, can be the gene. And it can be really interesting. Here, the case where we have my sequestrosome, my P62 gene that's found in a mouse, right? And it's indicating that you're using the top, well, pretend it's the top, but you're using one direction of the DNA, okay, as code. This MGAT gene, this MGAT4B gene, is not at the same place. It's further down, but it's going, instead of reading the DNA from this strand, it's actually part of the DNA that's down from this strand, okay? Now, compare that to humans, which have the same genes, right? And they are going in opposite directions as well. But look here, they actually overlap. The promoter region of sequestrosome would be out here about where the word sequestrosome is, which means it's part of this gene here. And this MCAT gene is actually, the promoter region of it is off this direction, which means it's right in the middle of sequestrosome here. So some of those introns that we talk about are actually parts of promoter regions for other genes. There's this gene overlap that occurs. And you can see here that those would probably overlap. And you've got all sorts of ways that you could have potential overlap. So the region here in sequestrosome, the region that's opposite of the actual gene up here is the region of regulation that controls whether this gene is turned on or off. And the region that's opposite of the actual gene over here, the transcriptional unit, is the regulatory region for controlling whether this gene is turned on and off. So you get to use both sides of your DNA for various things, and it plays a big role in how the function works. Just to kind of give you an idea, when we start looking at genes, it really does kind of become quite complicated as to where they're found. Right? Okay, well now we know what a gene looks like. We're going to go back and on the next series of lectures and talk about transcription and RNA processing. You've already been through transcription. We're going to go through it in a fairly cartooned way. I say you already have. I know you have from other general basic biology courses that you've had. You may have had cell biology or others, or even some of you may be dealing with biochemistry where you go into it in depth. We're going to go through it from a fairly simple standpoint where we're simply looking at how do we get this DNA made into message that we can use to make protein. Because remember, our whole process here is to go from DNA, the genome, to phenotype, which is protein in our case, right? And that's what we're going to look at next.